This is Hidden, a podcast about things unknown and people we are celebrating.
So I was an undergrad at WashU in St. Louis. I knew I liked environmental science. I was like being outside, flipping over rocks, looking for amphibians, that sort of stuff. And I got involved in a community ecology lab run by John Chase at the time. And Kevin Smith was also affiliated with the lab and recruited me to do an undergrad research project with him in the summer. And at the time, so not long before this, a infectious pathogen, chytrid fungus, had emerged in amphibian populations around the world, which was really intriguing to me. Kevin had worked on this pathogen in Africa previously, but no one had looked to see whether it occurred in Missouri.
What you are hearing is discussion regarding an infection, chytrid fungus, with amphibians in Missouri ponds.
And so the project that we worked on was to join along on an existing pond survey run by the lab at large. They're looking at amphibian communities, macroinvertebrate communities, and other species in these ponds throughout Missouri. And so my part of the project is on a subset of the amphibians that we collected. Kevin and I sent them off to find out whether they tested positive for this pathogen, the chytrid fungus.
The person we are discussing this with assisted with this research in 2013.
Hi, my name is Alex Strauss. I am an assistant professor in the Odom School of Ecology at the University of Georgia, and my lab studies the community ecology of infectious disease.
So I have a quick question about that.
The other voice you will hear is our co-producer, Tambora Mills, who assisted in this show.
I know that you said that he saw that it was present in South Africa. Why the jump to Missouri, which is quite a big distance away?
So the most famous, or infamous, I should say, declines in amphibian populations due to this pathogen occurred in Australia, Central America, Sierra Nevadas in the United States. So really all over the world is my point. It sort of very rapidly became apparent that this thing was everywhere, and that in some places it caused declines and in some cases extinctions. We've lost a bunch of amphibian species to this pathogen. And so there was a reasonable expectation that it might be in Missouri as well, and just no one had looked. So we wanted to know.
On this episode, we speak with Professor Alex Strauss regarding his research with parasites in disease ecology.
The chytrid fungus, I do know that they are of conservation concern. And I also know from our work that although chytrid is present in Missouri, or was present in Missouri 10 years ago, 15 years ago, oh my gosh, time flies. It wasn't associated with any. We didn't see any negative consequences. So from our work, it seems like it's probably been there for a while. Some species test positive. We were looking at leopard frogs and tree frogs, but no signs that their populations were declining in response to presence of the pathogen. and curiosity is what keeps me going. So that's the reason you get into science in the first place, I think.
I never got into science, but like I said, it was always one of my favorite subjects, and I love talking to people.
I would say that detecting the presence of this pathogen or of this species that has caused declines in other parts of the world, I mean, it's probably not great that we found it in Missouri, but it's also not necessarily a cause for concern. It seems like it has a global range at this point. It's everywhere. And that if it wasn't causing obvious declines, it might continue to not cause declines.
A subdiscipline of ecology, disease ecology is the understanding of the mechanisms, ecological causes, and patterns of course that impacts on host individuals, communities, and populations. This work is typically with quantitative approach.
I mentioned that I have sort of moved somewhat away from these ideas, but I'm still very much a community ecologist. So I'm interested in the way species interact with each other, how the environment changes those interactions, and how interactions between two species, say for example, predator and a prey, what that might mean for other species in the same food web. And the healthy herds hypothesis is the idea that predation might improve health outcomes for a prey population if the predator is selectively targeting and removing infected hosts. Whether that's maybe because they're easier to catch, easier to see, sort of like the culling the weak idea. So predators might actually benefit populations of their prey in that way. Still study community ecology. I'm interested in those species interactions. I am still very interested in parasites and the roles they play in ecological communities, the way that those species interactions affect disease transmission. Most of the work that I do now in my lab is on communities of zooplankton, so the bottom of the food web, especially in lakes and ponds. So they eat algae, then they're the food for fish.
Professor Alex Strauss has a lab at the Odom School of Ecology at the University of Georgia that studies conservation and community ecology.
And for the most part, the questions that my lab asks are, I'd say, put under the category of basic science. We're interested in basic processes rather than conservation concern or a specific applied interest. The one exception to that is with guinea worm parasites. So guinea worm is this truly horrifying parasite that infects people. And it alternates between infecting people and infecting a specific kind of zooplankton called copepod. So if you know SpongeBob, plankton from SpongeBob is a copepod. These little critters, little aquatic critters, and if a person drinks water that has an infected copepod in it, it can develop into an adult worm. There's multiple worms. They can reproduce, migrate through your body, grow to the size of like a 2-foot long spaghetti noodle. And then the horrifying part is when they move then to your extremity, typically like your foot. It creates this burning sensation. And as the worm then burrows to the outside, it creates the sensation of needing to stick your foot in water to relieve the burning, and that's how the parasite releases its eggs back into the water, where it can infect those copepods again to complete the life cycle. So really horrifying and also ancient human parasite. So the caduceus, the symbol that's on like an ambulance, that's the guinea worm parasite.
For decades, the late and former President Jimmy Carter worked to eliminate the painful parasitic infection, the guinea worm disease. In 1986, there were a total of 3.5 million cases reported.
So, fortunately, guinea worm parasites have almost been eradicated. They're down from thousands of cases per year to just a handful. But where it still causes infections in people, there's a few countries in Central Africa, and despite really substantial eradication efforts, really led by the Carter Center, Jimmy Carter's foundation, there's a few places where the parasite is just really hanging in there and really resisting these eradication efforts. And so my role in all of this, I'm relatively new to this much more applied type of research, but we're trying to learn more about the populations of those copepods, the zooplankton that are the intermediate host for the parasite, and especially how predation by fishes in some of these water bodies affects the likelihood of people getting sick with the parasite.
When you mentioned that you'd had an interest in that and that you were working towards that, it kind of brought up that I had watched a lot of videos about the Carter Foundation and this some years ago. And I remember it being truly horrific because, what you're studying a lot of times are these things you just can't see with the naked eye. There's a danger you can see, a clear and present danger, fires, bears, predators, you see those. And this is something that in these areas with, little to no sanitation and fresh water and things of that nature, that this would be, a huge thing. And to see the cases go down from millions to, I think it was 14 or 15 cases last year, is one, fascinating, right? And that says a lot about the efforts to try to eradicate it. But then when I saw, because I was thinking, I guess they created a drug to fix this, and I saw what you have to do to get the worms out and was horrified. If anybody who has not seen it, I would suggest going and watch a video if you don't have a weak stomach. But that whole life cycle, which you're talking about, kind of makes sense. And I guess your research is maybe early detection and or stopping the cycle so that it could, maybe not be a problem in the future in these places, or water treatment.
Yeah, it's about. I'm glad you brought that. So this idea that parasites are small but can be really important and have big consequences, that's honestly one of the things that got me into this area of research in the first place. And it's also, I think, maybe more salient with many of us after, the COVID-19 pandemic, and just, feeling that ourselves. So, the goal here is to help get over that last stage of these eradication efforts by learning more about what this parasite, how this parasite is surviving in the wild, in nature. It's not really nature; it's ponds that are visited regularly by people because that's how they get their water. But it also really puts into perspective the amazing, the things we take for granted in a more developed part of the world, where you want to drink water, you just go to the kitchen faucet and, fill your cup. You don't have to worry about, with some exceptions, of course, but, for the most part, we have a lot to be really grateful for. That the last step is, I think, going to be incredibly challenging. There's only 2 pathogens in the world that have ever been successfully eradicated. One is smallpox, and the other is rinderpest. This one that infects ungulates from the Serengeti. And I think that's a testament to how difficult it is that it's only been successfully completed twice in human history, despite generations of solid information and hundreds of probably maybe even thousands of pathogens that can infect people. The real challenge, and I mean everything you said is correct, that ongoing transmission is in more remote places. The Carter Center is doing an excellent job, I think, of learning a ton from those locations. But one of the things that has come to light in the last 10 years or so is that humans are not the only thing that can get infected by those adult worms. So more cases have been identified in dogs in particular, but also wild cats, and baboons, a few other species. And so that really throws a wrench into eradication efforts, because there's parasites that could be surviving, propagating, undergoing their life cycle by alternating between the copepods again and some other species where we have even less information, because the dogs aren't going to go report an infection when they get it, especially if it's a stray. So all of that is to say, I think that is re-emphasizing the importance of learning more about the copepod populations, because they are also one of the parts of the life cycle that the Carter Center has pinpointed and is putting a lot of resources into targeting to slow transmission. So there's a chemical called abate, which is applied to water bodies, also for mosquito control, for malaria and other mosquito-borne parasites. But one of these management strategies is to try to knock out the copepod populations, because then they can't transmit parasites to, people, dogs, baboons, whatever. And part of our research on this topic is to try to understand, when that works well, but also, if it doesn't work as well, what are some of the reasons for that? Again, this isn't specifically my area, but a single adult worm can release, I think, hundreds of thousands of eggs that hatch into infectious larvae. So, another example of why those last few are really challenging, because all it takes is one to prevent eradication from being successful.
And then, I guess from your part, you're at the beginning stages, and then up until it gets to the copepods. How big are we talking, or how small are the things that you're looking at? These the different I don't know what their larva looks like, but the zooplankton I know are super tiny.
Yeah, and so the two big categories of zooplankton are copepods and cladocerans. Most of my lab projects are about cladocerans. The exception being this one very applied project with the Carter Center on guinea worm transmission. But both the cladocerans and the copepods, so both these kinds of zooplankton, I mean, they vary in size. There's lots of species of both, so it's not, they're not monolithic. But anywhere from maybe like a tenth of a millimeter up to 2 millimeters. So they're small, but you can see them with a naked eye, which is really handy for being able to catch individuals, isolate individuals, to do experiments, monitor population growth, counting individuals over time. So they're small, but not so small you can't see them. The larva of the first instar larva of the parasite are small enough that they are eaten by those copepods. So they're much, much smaller.
2 millimeters is bigger than I thought you were going to say.
Yeah, 2 millimeters is right. You can totally see them. You can even count them. In fact, most of the population studies that my lab does, you don't even need a microscope. You need a microscope if you have multiple species in there and you need to identify, different things and count the numbers of each one. Or for a lot of our projects now, we're looking at stage structure, which means, within the population, what proportion of them are juveniles or adults, because that can actually tell you a lot about how the population is growing. So, if you want to know something about different species within that community of zooplankton or about their stage structure, then you need a microscope. But otherwise, you don't even need a microscope. You can just, it's easier if they're well lit, but then you just count them.
And then are they getting like water samples and everything and then shipping it over to you? Because you're in Georgia, correct?
Yeah, so I'm at University of Georgia. One of my collaborators on this project, Dave Civitello, has a lab at Emory, so also Georgia in Atlanta, which is also where the Carter Center is based, by the way. So Dave and his team have been going to Chad, working with people there. So most of the Chad-based field research is run by a local team. Dave and a couple other Emory people go every couple months, and I'm going to be going for my first trip sometime this spring, although I haven't yet worked out those details yet. I'm really looking forward to it. We've been doing a bunch of experiments, essentially trying to recreate different field conditions. So we're manipulating turbidity and mesocosms. A mesocosm, basically, you can think of it as like a big bucket, like an artificial pond, where we're trying to create conditions that are similar enough to field conditions, but under controlled enough conditions that we can manipulate particular variables that we're interested in. So we've been doing a bunch of that work, but I really want to be able to see these ponds myself to get a better sense of, what exactly it is that we're trying to recreate in the lab. I think the best ecology emerges from a combination of field studies. Like if you're not out in the real populations or communities that you're trying to understand, you're really limited in what sort of inferences you can draw. So, I think the best ecology is often grounded in field studies like that. But then the value of lab work and experiments is it lets you ask a very specific question. Say, for example, in this case, in the field, we have some preliminary data that indicates that ponds, I should say water bodies more broadly, so this includes ponds, rivers, really wells, really shallow pools. Those water bodies that have higher turbidity have much larger copepod populations. And they also they're much less likely to contain fish predators. And so that, suggests some intriguing hypotheses about maybe the turbidity is enabling the copepods to escape predation, or maybe the turbidity is preventing fish from being able to survive in the first place. In either case, those are hypotheses that we can test by manipulating turbidity in the lab and then, asking how that is affecting predation rates or population dynamics of the copepods. But then if we go out in the field, and see something completely different, then, like the whole idea of a hypothesis is rooted in this idea about our conception of the way we think things are operating in the field. And so, I'm hoping that this has been a, useful set of experiments we've done. But if we get out in the field and observe something unexpected, it could suggest a completely different direction to go. There's also another wrinkle here, which I think makes the academic ecologist in me gets excited about this. But there's this other potential transmission route where if a fish eats an infected copepod, the parasite isn't able to infect the fish, but it is able to survive. And so then if that fish gets eaten by a dog, specifically if the guts of the fish, where that parasite is maybe still alive, biding its time, get eaten by a dog, that could also be a transmission pathway. And so there's multiple ways here that fish predation could be influencing the probability of infection. Sort of this one, I'd say, classic sort of healthy herds type route where if you have predators, you're just going to have a smaller population of prey, which means fewer of them are going to get infected, which would be good. But the flip side is, right, if you get fish at the wrong time, they could be sort of consolidating all of the infectious copepods, getting all the parasites in their fish guts, and then if that fish gets caught by somebody and its entrails get thrown to the dogs, that could lead to infection of the dog. So part of the goal of our experiments is to try to understand better which of those two seems more likely. And I guess it's too early for me to weigh in more specifically on that.
Well, I, for one, am glad. I'm glad to actually talk to you and hear the passion in your voice. I mean, you can see it through all of your accolades when you look you up. It's a different thing to actually hear that kind of passion, that thirst for knowledge, trying to understand. Honestly, I mean, you probably don't hear it enough, but thank you for just being curious and trying to figure out what's going on, because I think that your research and all the things you do will help and have helped millions. So.
I will say that I think that this guinea worm project is really important. I've enjoyed working on it, but it's also a relatively small fraction of the way I spend my time. I mean, I shouldn't say small, maybe 20-30% of my time. And I think there's just an enormous amount that we as people, as ecologists, don't know about parasites in general. Not necessarily the ones that infect people. The ones that infect people get studied a lot more than the ones that don't infect people, as we can imagine. And yeah, I think there's so much to be curious about, so many reasons to go outside and observe nature and be open-minded, come back to the lab with specific hypotheses, ideas you want to test. And yeah, I love my job. It's a lot of fun.
Professor Strauss states there is still much to learn about parasites.
I have a lab website. It is strausslab.ecology.uga.edu. And I guess I'd say to listeners who are interested in this type of conversation, I would describe myself as a disease ecologist, and there's a lot of other disease ecologists out there. People who study disease ecology from applied perspectives, like that guinea worm project I was talking about, and then also people who study pathogens that infect plants, or species like frogs of conservation concern, or who just study parasites because they're interested in understanding more about parasites and what roles they play in nature.
We would like to thank Professor Alex Strauss for his interview. And special thank you to my co-host, Tambora Mills. This is Hidden is brought to you by Southside 2-Bit Studios.